Good day. I will discuss the description and principles of electrocardiogram. An ECG is simply a representation of the electrical activity of the heart muscle as it changes with time, usually printed paper for easier analysis like other muscles. Cardiac muscles contracts in response to electrical depolarization of the muscle cells. It is the sum of these electrical activity when amplified and recorded for just a few seconds that we know as an ECG. The basic principles of electrocardiogram is the heart generates by itself an electrical activity which is transmitted through all the organ and produces its contraction. The electrocardiogram is no other than the graphical representation of this electrical activity. Diagnosis of heart disease such as disease or conduction system alteration depends on the EKG interpretation. In addition, the electrocardiogram can be useful in the other anomalies such as the thromboembolism or electrolyte disorder. So electrocardiography is the process of producing an electrocardiogram. So a recording a graph voltage versus time of a electrical activity of the heart using electrodes placed on the skin. So these electrodes detect small electrical changes that consequences of cardiac muscles depolarization followed by repolarization during each cardiac cycle heartbeat. So the changes in the normal ECG pattern occur in numerous cardiac abnormalities including cardiac rhythm disturbances, inadequate coronary artery blood flow, and electrolyte disturbances. There are three main components to an ECG: the P wave, which represents the depolarization of the atria; the QRS complex, which represents the depolarization of the ventricles; the T wave, which represents the repolarization of the ventricles. So as you can see here, this is the example of an ECG paper. During each heartbeat, a healthy heart has an orderly progression of the polarization that starts with pacemaker cells in the sinoatrial node, spreads throughout the atrium, passes through the atrioventricular node, down into a bundle of his and into the Purkinje fibers, spreading down to the left throughout the ventricles.
The next recording device is the vector cardiogram. Vector cardiography, or VCG, is a method of recording the magnitude and direction of the electrical forces that are generated by the heart by means of a continuous series of vectors that form curving lines around the central point. In contrast with the electrocardiogram, which displays the electrical potential in any one single axis, vector cardiogram displays the electrical events simultaneously in two perpendicular axes. This gives a vectorial representation of the distribution of electrical potentials generated by the heart and produces loop-type patterns. Spatial QRST angle, or SA, is the angle of deviation between two vectors. QRS axis represents all of the electrical forces produced by ventricular depolarization, while T-axis represents all the electrical forces produced by ventricular repolarization.
Next recording device is electromyography. Electromyography, or EMG, is a technique for evaluating and recording the electrical activity produced by skeletal muscles. It is a diagnostic procedure to assess the health of muscles and the nerve cells that control them. It is performed using an instrument called electromyograph to produce a record called electromyogram. EMG works by detecting the electrical potential generated by the muscle cells when it is electrically or neurologically activated. The results can reveal nerve dysfunction, muscle dysfunction, or problems with nerve-to-muscle signal transmission. These signals can be analyzed to detect medical abnormalities, activation level, or recruitment order, or to analyze the biomechanics of human and animal movement. For equipment and parts of the EMG, electrodes, a high-gain amplifier, and an arrangement for recording the output. There are two kinds of EMG, surface EMG and intramuscular EMG. Surface EMG assesses muscle function by recording muscle activity from the surface above the muscle on the skin. Surface electrodes are able to provide only a limited assessment of the muscle activities. This can be recorded by a pair of electrodes or by a more complex array of multiple electrodes. More than one electrode is needed because EMG recording displays the potential difference or voltage difference between two separate electrodes. And then intramuscular EMG can be performed using a variety of different types of recording electrodes. The simplest approach is a monopolar needle electrode. This can be a fine wire inserted into the muscle with a surface electrode as a reference, or two fine wires inserted into a muscle reference to each other. Most commonly fine wire recordings are for research or kinesiology studies. Diagnostic monopolar EMG electrodes are typically insulated and stiff enough to penetrate the skin, with only the tip exposed, using a surface electrode for reference. And a high-gain amplifier, which is 10 to 5000 Hz, which is connected to an oscilloscope. Oscilloscopic traces may be photographed or stored on magnetic tape. Signals may be fed to an audio unit for on-the-spot feel of the signals. Lastly, an arrangement for the recording of the output. EMG is best done on a specially constructed shielded room to prevent interference. Procedure for EMG. First is one or more small needles or electrodes are inserted through the skin into the muscles, and then an electrical activity picked up by the electrodes is then displayed on an oscilloscope, and an audio amplifier may be used so the activity can be heard. EMG measures the electrical activity of muscles during rest, slight contraction, and forceful contraction. The action potential, size, and shape of the wave that is created on the oscilloscope provides information about the ability of the muscle to respond when the nerves are stimulated. As the muscle is contracted more forcefully, more and more muscle fibers are activated, thus producing more action potentials.
Hello everyone, I'll be talking to you about phonocardiography, which is also known as PCG. This picture shows different types of PCG. One is the old type and the other one is connected to a smartphone. Before I tell you what PCG is, I'll define you first what auscultation means. Auscultation is the technique of listening of sounds produced by the organs and vessels of the body. The area at which heart sounds are heard better is known as the auscultation area. It is important to know what auscultation is since PCG follows under this principle or technique. For the process of graphical recording of heart sounds or murmurs is known as phonocardiography or PCG, in which it uses an instrument used for recording sounds connected with the pumping action of heart known as a phonocardiogram. Phonocardiogram is a high-fidelity recording that represents the rhythmicity of the heart and, of course, its heart rate. There are two categories in order to record the sounds connected to the pumping of the heart. One is the heart sounds and the other one is heart murmurs. Heart sounds are transient in character and are in short duration. It has low frequency and it is mainly due to closing and opening of the heart. Mechanical working processes of the heart produce sound which indicate health status of the individual. So, the opening and closing of the heart valves is due to the relationship between blood volumes, pressures, and flows within the heart. Normal heart sounds loop and loop, which occurs during the closure of the valves. The other category is the heart murmurs, in which it is noisy and has higher pitch sound and are in long duration. It has higher frequency. It has also a high frequency. Murmurs are extra heart sounds that are produced as a result of turbulent blood flow in the heart and large vessels, which is sufficient to produce audible noise. Innocent murmurs are present in normal hearts without any heart diseases. On the other hand, pathologic murmurs are as a result of various problems such as narrowing or leaking of valves or the presence of abnormality. Some instrumentations are needed in order to create an effective phonocardiography. Uh, a phonocardiograph consists of a basic transducer in which it consists of a piezoelectric sensor that converts sounds or vibrations into electricity, and a crystal or moving coil microphone having frequency response between 5 Hz and 1000 Hz. Another one is the amplifier, in which it amplifies signals and offers selective high-pass filter to allow frequency cutoff. Next is the filter, in which it avoids aliasing and permits selection of suitable frequency bands. It separates louder frequency signals from lower intensity and much informative high-frequency murmurs. Next is the integrator, in which recording envelope of higher frequency over 80 Hz along with actual signals below 80 Hz. A power amplifier increases the power of incoming signals and it is more efficient. The effect, uh, the noise in here is also lowered. DACs and readout or high frequency chart recorders or oscilloscope or headphones are used in order to convert signals to digital form and stored permanently. It is also used for faithful recording of. This shows how BCG works. Of course, first you have your sensors placed in a patient's chest, and with the use of microphone, the uh, the sound or vibrations is converted to electricity, thus going to the IC recorder. And in order for us to see the wave waveforms, the wavelengths of the heart or the activity of the heart, the IC recorder is connected to the computer, or we can also hear our own heartbeats using the earphones. The last slide also shows the diagram of how BCG works.
The last recording device is electroencephalogram. Electroencephalogram, or EEG, is a non-invasive method for measuring brain waves of a person. It measures and records the electrical activity of the brain over a desired period of time. EEG can determine changes in brain activity that might be useful in diagnosing brain disorders. It works by having a technician that puts about 20 small sensors on your scalp. These sensors, called electrodes, pick up activity from cells inside your brain called neurons and send them back to a machine where they show up as a series of lines recorded on moving paper or displayed on a computer screen. It is helpful for diagnosing or treating the following disorders, especially epilepsy or other seizure disorder, or like brain tumor, brain damage from head injury, brain dysfunction that can have a variety of causes, inflammation of the brain, stroke, or sleep disorders.